Good evening, Pratham. Okay, so I'm Rahul Saha, and today we'll be progressing our talk on the photosynthetic pathway. In the last class, I had described to you the, you can say, green sulfur bacteria, purple bacteria, and also the Z scheme, the cyclic and non-cyclic photophosphorylation. So today we'll be continuing our talk, and we'll be talking about the dissipation of energy at heat, as you can say, dissipation of energy as heat, does it occur, and also the specific photosystem PS1 and PS2, and how do they prevent the excitation of both the PS1 and PS2 together, and how does PS1 always not get excited. Okay, so the thing is, when we talk about the, you can say, thermodynamic factors regarding the dissipation of energy. Okay, so just to give you a perspective, okay, of thermodynamic factors. regarding dissipation, okay, regarding, you can say, dissipation of energy. So what can happen is, regarding dissipation of energy. So what can happen is, the complex construction of the reaction center, okay, is a great product of, you can say, evolutionary selection for efficiency in the photosynthetic process. So if you talk about bacteria, right, the purple bacteria, the excited P780, okay, the excited P780, good evening, Samist. So the excited P780 could decay to its ground state by simple mere, you can say, internal conversion, okay, which is a very rapid process. If that process ever occurred, you can go to or reach P780 decay quickly. It will not be transferring the electrons to anywhere, it will transfer directly to its own ground state, and that decay would take a mere time of only 10 picoseconds. 10 picoseconds, which is 1 picosecond is 10 to the power minus 12 seconds. But still, the reaction centers are constructed in such a way. I had explained to you the fundamental concept of fret, that they are 10 meter or less apart only, that they are constructed in such a way that the inefficiency that this is 10 picosecond decay is completely, or you can say maximally, prevented. Okay, so this accounts for the high efficiency and rapidity of the reactions. So nothing is left to chance collision or random diffusion. The exciton transfer from one antenna chlorophyll to the special pair of the reaction center is accomplished in less than 100 picosecond. So entire thing is accomplished in less than 100 picosecond with an efficiency of more than 90%. Okay, so antenna chlorophyll to photochemical reaction center. This entire process takes, you can say, only 100 picosecond. Okay, only and only 100 picosecond with more than 90% efficiency. Okay, always remember this fact, more than 90% efficiency. Okay, so now coming on to, you can say, the next part, like within 3 picosecond of excitation, you can think of that pheophytin, which is the chlorophyll lacking the magnesium ion. It is able to, you can say, get reduced, and it has received an electron and become a negatively charged radical. Okay, and less than 200 picosecond later, the electron has reached the quinone B, or QB, you can say, right, ubiquinone. So it is a, you can say, very rapid transport of electron from one specific entity to the other, or one electron carrier to the other. Okay, next is, in plants, the two reaction centers act in tandem. We know that the two photosystems have complementary and distinct function. Now talking about the photosystem II, which we had observed and we had discussed the electron transfer through photosystem II, that is dependent on the wavelength of 680 nanometer. So that is known as PH2. Now PH2 is a, we know that it's a pheophytin quinone type of system. Okay. Now it resembles the single photosystem of purple bacteria, just like the P870. Okay. If we talk about PH2, So it's a pheophytin quinone type system. Then what it does is it resembles the purple bacteria in its function. Okay, means non-cyclic, the purple bacteria P870. Okay, that I had explained to you, this is non-cyclic photophosphorylation. Okay, only ATP will be produced. Okay, only ATP is produced. Next is in photosystem 2, it contains roughly equal amount of chlorophyll A and chlorophyll B. Very, very important. Contains roughly equal amounts of chlorophyll A and chlorophyll B. Okay. Next is, we know that the electrons are driven through the cytochrome B6F complex with the concomitant movement of protons across the thylakoid membrane, okay, into the stroma, right? And because the proton movement is just the opposite compared to the mitochondrial transport, which I had explained to you in the last class only, that in case of plants, we have thylakoid membrane, so it will be transported in this direction, okay. And in case of the mitochondria, protons are transferred in this direction, like this, through the, you can say, complex 1. Okay, so it is just like B6F, it resembles the complex 3, okay, and protons are pumped. After that, we talk about just the direction is different of proton pumping, but the electrochemical gradient will again be established. Now, PS1 is structurally and functionally related to, you can say, type 1 reaction center of green sulfur bacteria. Okay, it can go for both cyclic and non-cyclic. Okay, photosystem 1, PS1, structurally and functionally related to related to, you can say, the type 1 reaction center the type 1 reaction center of green sulfur bacteria. Okay, now it goes for cyclic and non-cyclic both. Okay, goes for cyclic and non-cyclic both, as we had discussed in the last class, that from ferredoxin it can return or traverse the electron back to cytochrome B6F, which will lead to ATP production of, you can say, the same ferredoxin, which is the iron sulfur protein. It can translocate the electron directly to the FD NADP plus, you can say, reductase generating NADPH, right? So it is both cyclic and non-cyclic. Then one more thing I'll tell you is very important, that it contains a high ratio of chlorophyll A to chlorophyll B, means more chlorophyll A is present, not equal. More chlorophyll A is present in PS1. Okay, so high ratio of chlorophyll A to chlorophyll B is present in PS1. Okay, now these two reaction centers in plants, they act in tandem to catalyze the reaction driven movement of electrons from H2O to NADP plus. We have known this altogether. Again, electrons are carried between the two photosystems. I have told you in the last class also, by the soluble protein known as plastocyanin. Okay, soluble protein known as plastocyanin, water soluble. Okay, water soluble protein that carries electrons from one photosystem to the other. Okay, so this is what happens in case of, you can say, plastocyanin. Now, entirely what happens is, we know that I had explained to you in the last class that when you go for electron transport chain, so I'll just share that image with you. When we go for electron transport chain, wait, I'll just share that image. Find it. Okay, I'll share it right now. Okay. So now it will be visible in few seconds to you, that is specific image. Okay, now you can see the image. Now in this image we can see that, we can behold that the electron that has been lost in P680 is not regained by P680, whereas electron lost by P700 is regained by P700 by the same electron that travels through the pheophytin plastoquinone, okay, of the photosystem II complex. It is regained, but this one is not regained. So if this keeps on, this process keeps on happening incessantly, then what will be the ramification? The consequence would be that P680, all the atoms present in the P680, the chlorophyll atoms would be completely oxidized, and then thereby no more P680 can be oxidized, because at that same wavelength, if you have ionized it once, we know that the second electron or second ionization enthalpy is always more than the first. Okay, so that may not be efficient, or then that may not be sufficient enough equal to h nu to throw out another electron, cause the photoelectric effect there. This is nothing but a photoelectric effect, right? You are throwing the electron out from the outermost electron in the gaseous state. Okay, this kind of photoelectric effect only, right? Okay, compare this with h nu naught. You need a specific frequency equal to h nu. That nu must be specific frequency having a specific threshold frequency known as nu naught. Then only the electrons will be thrown out from the photochemical reaction center. Okay, so that may not be enough, and then this process will be ceased. This specific non-cyclic process will be ceased. Cyclic can still continue, but non-process will be ceased. Even if cyclic continues, this electron will not be able to suffice the cyclic 2. So this entire process will be ceased, because P700 will not get the electron back, or will not get the, or will not be able to gain the electron, which comes from where? From the P680 or complex 2 only, okay, or photosystem 2 only. So the problem here is what you have as how to replace this electron. So this electron is replaced by water. Okay, this electron is replaced by oxidation of water. So I told you, in case of green sulfur bacteria, oxidation of H2S or hydrogen sulfide occurs. Okay, but in case of the higher plants, the oxidation of water occurs. Okay, oxidation of water takes place. Now oxidation of water will only happen, okay, and oxidation of water happens, because of that only we sow the plant with water. Okay, we provide water to the plant, and that is the main reason water is needed by the plants. Again, like oxygen is the best electron acceptor at complex 4, water is the best electron donor to the, you can say, PS2. Okay, so oxidation of water will happen. Okay, it will lose electron, so that is done. So we know that in case of, you can say, photosynthesis, that water evolving complex, it is known as, it plays a great role. Okay, so before that I'll tell you more about pH2. Okay, so pH2, what we talk about when we talk about pH2, okay, pH2 is dimeric. Okay, photosystem 2 is dimeric. and each monomer is a huge protein. It's a huge complex made up of, you can say, 19 proteins or polypeptide chains. Okay, so very very hard to if you can say imagine. Okay, PH2 is a huge dimer made up of two basic subunits, whereas each monomer is made up of 19 proteins. Okay, so huge is PH2. Okay, photosystem to 19 proteins, you can think of how big it is. Okay, it has the reaction center P680. Okay, it has got chlorophyll binding proteins also. Okay, and carotenoids also, non-heme iron also, and one more very very special thing, an inorganic complex known as Mn4CaO5. This will come in very handy when I'll explain this class only the water splitting complex to you. Okay, and surrounding PH2 are additional chlorophyll binding proteins and also the light harvesting complex proteins. Okay, so that is there. And what happens is when the excitation happens in P680, okay, it produces P680 star. It has got ionized. Okay, it is an excellent electron donor. So within picosecond, it transfers an electron to pheophytin and provides it one negative charge. And with the loss of its electron, P680 star is transformed into a radical cation. Okay, radical cation. So P680 star transforms to a radical cation P680 plus. Okay, that is known as radical cation. So I'll write it down here. The loss of one electron, this P680 after ionization gets transformed to becoming a radical cation. Very very important. This is okay. And then the pheophytin which has accepted the electron, okay, it rapidly passes the extra electron to the protein bound plastoquinone PQA, okay, which in turn passes this electron to another loosely bound plastoquinone PQB. Okay, like this two processes happen. Okay, two times the same process will happen. Okay, P680 one electron gone, it will throw out or release the electron to pheophytin. Pheophytin will become negative. It will transfer it to PQA. It will transfer it to PQB. Okay, PQB will become a radical. Okay, so PQB will become a radical. Okay, and one more time, one more atom will be again, you can say, oxidized P680, another P680 star. It will become P680 plus. Again same thing. Now two electrons when it will get, okay, then PQB, and two protons, it will become PQBH2. Then it will become PQBH2. It will take in two protons, okay, and when PQB has acquired two electrons, in such two electron transfers from PQA, okay, and two protons from the solvent water, okay, then it becomes plastoquinol B, PQB. Okay, so that is the function of, and then PQB will pass it down through the cytochrome B6F complex, okay, which will cause then the proton gradient, proton pump it is, and which will produce the electrochemical gradient, chemiosmotic theory, which has been described to you earlier only, and then the protons will go out of the thylakoid membrane through the ATP production. You can say F1, F0, ATP, AS protein, transmembrane protein, complex 5, and then in humans, and then the process will continue. Whereas photosystem 1 is not a dimer, okay, it is just made up of 16 proteins, okay. Photosystem 1, if you talk, PS1, it is not a dimer, it is just made up of 16 proteins, okay, much smaller in comparison to, you can say, photosystem 2, and it also includes chlorophyll binding proteins and carotenoids, okay, then FES cluster that we have talked about in the biotech class, okay, so those things are still present here to carry the electrons in PS1, okay. Now what happens is the specific cytochrome B6F complex, okay, is very, very essential. When you talk about the cytochrome B6F complex, okay, it is just like the complex 3 of mitochondria. So complex 3 will be taught, okay, in biotech very, very soon. Okay, in the electron transport chain you will be able to understand how, why I am talking about complex 3 and all. Okay, so it is a great electron pump and is analogous to complex 3. When I will teach complex 3, it will become very, very clear what is the meaning of, you can say, cytochrome B6F complex and how it is related to complex 3. Okay, now one more thing, the cyclic electron flow between PS1 and cytochrome B6F complex, okay, it will always, you can say, if the cyclic flow happens, what will happen? It will always increase ATP production. So that is the, you can say, perfect rule of this specific, you can say, pathway. One more thing I'll ask you is, plants are always exposed to light of highly variable intensity and wavelength in the course of a day or a season. Okay. And although they can alter their growth patterns somewhat, they cannot uproot themselves and move to optimize their light exposure. Okay. Instead, what they have is they have the, you can say, cellular mechanisms which have evolved that allow plants to accommodate to changing light conditions. Now you see the energy needed to excite PS1, okay, is 700 nm, a longer wavelength. Energy, just put your concentration here, that energy needed to excite PS1 is 700 nm, which is a longer wavelength, right? Longer wavelength than, you can say, PH2, which is 680 nm. And we know that E is equal to h nu. Longer wavelength means higher frequency. So whose energy will be higher? PH2 energy will be higher than PS1 energy. So where will you need to put more energy to excite electron, PS1 or PS2? You will need to put more energy to excite electron in case of PS1. Now, the, you can say, if PS1 and PS2 were physically in contact in the chloroplast, like physically contagious, the excitons originating in the antenna system of PS2, okay, would easily migrate to the reaction center of PS1, not PS2, because it is easy to go to a lower energy than go to a higher energy, right? So it would require higher energy, so this will take the route of the lower energy, it will pass down to the reaction center of PS1, leaving PS2 reaction center chronically or always under excited, okay, and thus it would never be able to cause the non-cyclic flow. Okay, non-cyclic flow will not happen, PS2 will never be activated, and if PS2 is never activated, okay, PS1, the P700 will also not get electrons. Okay, always remember that it will also be terminated automatically after one cycle. Okay, or till all the atoms are oxidized, okay, for P700. So the thing is, if pH2, I'll write it down for you, if pH2, this is a very, very critical scientific question, okay, for you to ponder, were physically contagious, okay, what would happen is excitons originating, okay, excitons originating, you can say, in the antenna system of pH2, antenna system of PH2 would do what? Would migrate to the reaction center of PS1. Okay. Oh, good, Samist. Good. So there is still scope for improvement, and most of those things I don't think the planned part was taught by me. So still you have done good. Okay. Appreciable marks. Good enough only, because in my paper also people get highest marks are like this only. Okay, so if you keep that up, you can get more than 16 annual examination. Okay, no problem. So the thing is, in the antenna system of PH2, okay, where I was, yeah, in the antenna system of PH2 leaving reaction center of PS1, leaving the migrate to the reaction center of originating in the antenna system of PH2 would, yeah, would migrate to the reaction center of PS1, leaving, you can say, PS2 chronically underexcited, and if it is chronically underexcited, what will happen? And thus interfering with the operation of the two center system, and thus interfering with the operation of the two center system. Okay, so this would happen. Now how is this circumvented? Okay, as per quantum mechanics, E equal to h nu, only P700 should get excited because it has got lower energy. Now how would you cause the excitation of PH2? Okay, that is why evolution again comes into action. Now what happens is, PH2 has, on PH2 and PS1, they have been separated in location inside the chloroplast only. Okay, so the supply of excitons is prevented by, you can say, separating the two photosystems in the thylakoid membrane. PH2 is located almost exclusively in the PH2 is located almost exclusively in the, you can say, tightly appressed membrane stacks of granule thylakoid. Tightly means they are located in the membrane of the granule thylakoid. Appressed membrane of, or membrane stacks of granule thylakoids. Then PS1 and its associated light harvesting complex, okay, LHC2, the light harvest complex protein, mediates the tight association of adjacent membranes of the grana, because grana has many membranes like this, so they are associated by LHC2, light harvesting complex 2, specified proteins. Okay, light harvesting complex 2, you can say. Nothing but these are proteins, okay, LHC2. Now where is the location of PS1? Okay, PS1 and also the ATP synthase, okay, PS1 and ATP synthase, which is the F1 F0 ATP synthase in case of, you can say, humans, okay, same analog is present in plants also. This complex is located almost exclusively in the almost exclusively in the non-appressed region of stromal thylakoids. To see how great, how enthralling the evolution is. Stromal thylakoids. Okay. Now, stromal thylakoids are the region where they will have the access to the contents of the stroma, including ADP and NADP plus. Okay. The cytochrome B6F complex is also primarily present in the granule thylakoid. Okay, as far as competitive exam is concerned, you can write down that the cytochrome B6F complex is also present in the granule thylakoid. Okay, also present in the granule thylakoid. Always remember this. Now, what happens is, I'll just tell you a straight transition theory, okay, just for your knowledge. Keep it for your knowledge only. It won't be asked in the examination. I'll just tell you for your knowledge two things that will make the concept very, very strong. That what you have is the association of light harvesting complex. Okay, this light harvesting complex, when it associates with PS1, PS1 is active. When it associates with PS2, PS2 is active. So in state 1, okay, light harvesting complex, okay, is associated with PS2. In state 1, this light harvesting complex is associated with PS2. Why? Because light harvesting complex, there is a specific residue known as threonine amino acid. That threonine amino acid is unphosphorylated, means there is no phosphate group present. Okay. So this phosphorylation and unphosphorylation concept comes a lot in the chapter cell cycle, right? Where you have cyclin, CDKs, CAC, and CKI. Okay, when you cross the checkpoints. Okay, so those questions are asked in NEET examination. So threonine is unphosphorylated. That's why threonine is one of the amino acid at the specific number that is unphosphorylated. So LH2 is associated with PH2. Now what happens is, under conditions of intense or blue light, okay, it will favor PH2 because you have a lot of energy. Okay, so now PH2 is activated and a lot of plastoquinone is being reduced since PH2 is active. So PQH2 is being formed. Okay, but if PQH2 formation, okay, PQBH2 formation is becoming a lot more, is happening at an enhanced rate, and this PQBH2, the electron must go to site of, you can say, the complex, sorry, the photosystem 1 P700, and then it must be, you can say, oxidized. Okay, later on PQBH2 must become PQB and it should transfer the electron to P700. But if P700 is inactive, only PH2 is active, since LC2 here is bound to PH2. So what is the result? This PQBH2 is getting accumulated and not getting oxidized. So it is not getting oxidized at the rate it is being produced. Okay. At the rate it is being produced. So what will it do now? That PQBH2 will activate a specific enzyme, a specific kinase, okay, now which will phosphorylate the LHC2 at its critical threonine residue. The phosphate group will be added because kinases take the phosphate from ATP, okay. That's why AS destroy the ATP means break the gamma phosphate bond, ADP plus phosphate PI, and this energy is utilized to catalyze the phosphodiester, sorry, the phosphoanhydride bond, covalent bond, this bond, this is that bond formation, and phosphate between the amino acid and this same phosphate. So that is what kinases do. They phosphorylate the compounds by using phosphate group from ATP. Okay, from where will they find the phosphate group? From ATP. Now, once this LHC2 has been phosphorylated at the threonine residue, now it will change its conformation and dissociate from PH2 and associate with PS1. When it associates with PS1, now PS1 LHC2 associated, now PS1 is active, and now it is able to reduce PQBH2, okay, because sorry, it is able to oxidize PQBH2 to PQB. It is able to now oxidize whatever PQBH2 has been generated. That PQBH2 will be converted to simple PQB, as P700 is now active, that pathway PS1. Now what will happen? PQBH2 level will go down. PQB is too much, PQBH2 is not present only, and then PQBH2 is lesser. So who will activate the kinase? Nobody. And then again, when that PQBH2 concentration is lesser, this kinase cannot be activated, then this will become dephosphorylated, and again PH2 will become activated. So this cycle keeps on going. Okay, this cycle keeps on happening this way, that way, this way, that way. Then one time PS1, one time PS2 is active with complete regulation. Not one of them is active for a very long period of time, and in the optimal state they remain activated. Okay, so this is how the activation of the entire, you can say, PS1 PS2, okay, it relates, and neither both of them are not really activated at one go. Okay, so now we'll be talking about the water splitting complex. Okay, now about 3 billion years ago, this is the very important topic, water splitting complex. So about 3 billion years ago, what happened was, billion years ago, evolution of primitive photosynthetic bacteria, okay, which are the progenitors of the modern cyanobacteria or blue-green algae. They produced a photosynthesis, you can say a photosystem capable of taking electrons from a donor that is available always, which was water. Now, if you apply basic chemistry here, so two water molecules are split, yielding 4 electrons, 4 protons, and 1 molecular oxygen or not. So 2H2O will give you 4 protons, 4 electrons, and 1 molecular oxygen. Okay. Now, a single photon of visible light, listen to this very carefully, a single photon of visible light equal to h nu does not have enough energy to break the bonds in water. Okay, so 4 protons are required in this photolytic cleavage reaction. In order for this cleavage to happen, 4 photons are required. So this is photolytic cleavage, meaning that photon associated or photon induced cleavage. Photolysis, lysis of H2O. Okay, photolysis slash photolytic cleavage. Photolytic cleavage. Okay, 4 protons are required to break this specific bond. Now, the 4 electrons which are released or abstracted from water, they do not pass directly to P680 radical. Note this down. Okay, very, very important. The 4 electrons, sorry, that has been abstracted from water, okay, does not bind to or associate with the P680 radical, okay, the cation radical. Okay, what happens is P680 cation radical can accept only 1 electron at a time. That is the reason, because this P680 can accept only 1 electron at a time. Okay, so instead, so you cannot just transfer 4 electrons, okay, and don't want to waste the other electrons, right? You suppose you are transferring only one, you are wasting three. Why do you want to waste three when 4 photons have been used up? Okay, and water is not an, you can say, unlimited quantity. Some people don't put water on in the soil. Okay, so you need to conserve it also. So the thing is, instead, a remarkable molecular device, the oxygen evolving complex or water splitting complex, has been developed by evolution in plants. This passes 4 electrons one at a time to P680. Doesn't cause or doesn't lead to the loss of or doesn't lead to occurrence of any loss of electron. Okay, one at a time to P680. Now the immediate donor to P680, okay, is what is a tyrosine amino acid which is present in PH2. In D1 you can say subunit, but at your level just think of tyrosine is present. It's a protein, it's an amino acid which is a component of protein present in the PH2. Okay, so immediate transfer is done by tyrosine which is present in PH2. Okay, what happens is when 4 times P680 means 4 atoms of P680 has been or have been ionized, 4 times PH2 has run, 4 atoms have become P680 cation radical. To this, 4 tyrosine amino acids will react. Okay, tyrosine amino acid will provide it electron, it will become P680 normal, and this will become free radical. Okay, tyrosine free radical. Okay, 4 tyrosine, each tyrosine will become a free radical. Okay, so this is the reaction number, you can say 1, or reaction number 2 you can write, because 1 is the that reaction, H2O breakage, photolysis. So this is reaction number 2. Now the tyrosine radical, it will regain its missing electron from where? I told you PH2 has got Mn4CaO5 cluster. Okay. Now that cluster will provide 4 single electron transfers to each tyrosine. Now 4 tyrosine will react with the Mn4CaO5 whole 0 cluster. It will provide 1 electron to each tyrosine individually. It will become 4 tyrosine which have been converted from free radical to non-radical now. But this Mn4CaO5 has lost 4 electrons, so it will become 4 positive or not, right? This is also present in PH2 only. Okay, inorganic cluster in PH2. Okay, inorganic cluster in PH2. Now if you add that equation 1 with equation 3, this is equation 3, equation 1 was 2H2O photolysis equal to H nu 4H plus 4E minus plus O2. Just add equation 1 and 3. What you will get is, just add equation 1 and 3. You will get is Mn4CaO5 4 plus whatever you have got here, Mn4CaO5 4 plus 2H2O, okay, and this will lead to, you can say, 4H plus 4 electrons, okay, will reduce it to, you have 4 electrons here, it will reduce it to Mn4CaO5 0 again, whole 0, charge is 0 again because it has got 4 electrons from here. What is remaining is 4H plus is remaining and 1 O2 is remaining. This is why, this is the reason why. This is the reason why plants are able to evolve or release O2. That is why it is also known as oxygen evolving complex, water splitting complex or oxygen evolving complex. So now you can tell anybody, explain it to anyone, the concept behind oxygen evolution or the liberation of oxygen from plants. We all say that plants give out oxygen. How? This is how. Plants need water. Why? This is why. They need water. This is how they liberate oxygen. And this 4H plus will be pumped into the thylakoid membrane to increase the, you can say, electrochemical gradient. So this is not a waste. This will increase the electrochemical gradient, which will then be used up in the production of ATP later on. Okay, so this is your water splitting complex. So I hope today's lecture was clear. We were able to comprehend it. If you had any queries or doubts, you can contact me on my personal number. Okay then, thanks a lot. Let's meet you in the next lecture.